I think I will bump back to just as a little bit of review. I didn't like put together formal questions to refresh from last week, but I did pull forward some slides. So let's start with just kind of going over features of lymphocytes that would be also applicable to today's discussion about B cells. Okay. So a quick outline of things we need to cover. We'll talk about the humoral immune response, the adaptive humoral response, B cell mediated. And then at the last part of the lecture, we'll talk about some immune dysfunctions and talk about vaccines and how they work as well. So before we get into this, let's just skip back, review, recap some features of lymphocytes. So first of all, what are the lymphocytes? What cell types are they? White blood cells such as T cells and T cells. Yeah. And they are of what lineage? Are they myeloid or are they lymphoid? Lymphoid. Yes. Okay. So B cells and T cells, we are talking about primarily an adaptive immune function, meaning primarily, first and foremost, they are antigen specific. The function is antigen specific. We talked about T cells and how they have their T cell receptor on their surface that is exquisitely specific for one particular antigen type that's presented to them on the surface of antigen presenting cells or nucleated cells, depending on which, on this major histocompatibility complex on that surface is presented to them. With B cells, also antigen specific, also presenting a receptor on its surface, but it's a B cell receptor, and the shape is different. What's the shape like of a B cell receptor?
Antibody?
It's basically an antibody. Yeah, it's an antibody that instead of being soluble or free floating, it is bound in the membrane. So like here's your B cell, nice round sphere. You have the B cell receptor bound on the surface. So these little receptor areas, there are two of them, think of them as arms, which are pointing outwards away from the cell, ready to bind and interact with different antigens. We also talked about, in the context of T cells, about B cells, longevity and clonal expansion. The ability to recognize and kind of positively select for lymphocytes, like B cells, that recognize a specific encountered pathogen and then expand those populations so that you have a really big fighting force to eliminate or remove that threat. With B cells, like I said, you have these little baby B cells, which are fully functional, but they haven't yet encountered a pathogen yet. But they still have that BCR on the surface ready to encounter an antigen. Some of them may never meet that antigen, and others will. Others will encounter that. Where would this encounter probably take place? Where are these B cells going to be, these mature B cells with their BCRs? In the lymph nodes, primarily, yeah, the secondary lymphoid organs. So mostly in the lymph nodes. Could be the spleen, could be some of the mucosal-associated lymphoid tissues. But that's where they hang out once they've maturated. And unlike T cells, they don't have to go to thymus stool. They go straight from the bone marrow where they're developed and they're born to those secondary lymphoid organs. So they go to lymph nodes. So there they wait. Luckily, in the context of a lymph node, what are they interacting with? They're mingling with antigen-presenting cells and T cells. So if there's an antigen to be encountered, that's where that meeting is going to occur. And like I said, once they do meet their specific antigen, there are signals that tell those B cells that they need to proliferate, they need to expand, and they need to further differentiate into either plasma cells, which are the cells that start secreting soluble antibody, or memory cells. So a small portion of those will be memory cells. And the memory cells serve to basically not do a whole lot until or unless they meet that same antigen from a pathogen again. And then they have to get stimulated to start differentiating back into plasma cells at that point. But otherwise, they can just go about their merry way doing very little at that point. And lastly, that other feature that's important for B cells as well is immune tolerance. Specifically with those antibodies that are produced, those antibodies should not be binding to self, host tissue. They need to be binding specifically to pathogens. Otherwise, again, you kind of get the same issues that we talked about with T cells. You get autoimmune disorders and other problems. This very popular slide about lymphocyte surface markers. We talked about some of the T cell surface markers that were important. B cells too, I think we glanced through this stuff very quickly, but we had the BCR itself, the BCR, which was comprised of these CD markers, CD19, CD20, CD21. And what does CD stand for again? Clusters of differentiation. Outstanding. Yes.
Yeah.
So yeah. So CD19, for example, in that BCR complex, that's the one that if you're doing flow cytometry, for example, on patients' DMC or whatever, and you find CD19, those are mature B cells. It's a marker for mature B cells. They also, because they're antigen presenting cells, and we remember that from that dumb little acronym, they express MHC class 2 on their surface as well. So they are able to receive antigens and have an effector function for that as well. And then lastly, they have CD40 on their surface, and that helps them communicate with T helper cells when they're communicating with T helper cells, and receives important signals that tell them that they need to be activated and they're basically on the right path. They have the right antigen specificity on their BCR. Again, B cell receptor, there's CD40, and like any APC, any antigen presenting cell, you have your MHC class 2 on the surface as well. So that's very important surface markers for B cells. Okay, so that's all review material. Any questions on that stuff? Okay, so moving along, some of this we've already said, but this is an important slide. We know that B cells will develop in the bone marrow, and by the time they leave bone marrow, they will have that unique B cell receptor already formed. So it may not be selected for, but it leaves bone marrow with that BCR. So those are mature BCRs, and they have that CD19 on their surface. They go to those secondary lymphoid organs like the lymph nodes, and there they wait. They wait until they encounter a specific antigen, and that 1 in a million specific antigen that will bind to their BCR. So in this figure right here, this sort of mirrors what I'm talking about. But this FDC, that's a follicular dendritic cell. It's all those tentacles coming off of it. That dendritic cell is basically sharing an antigen with that B cell. So that B cell has the BCR receptor on its surface. It's receiving that antigen, and then the way it receives it is by receptor-mediated endocytosis. So that entire BCR/antigen complex will be internalized into that B cell, and then it goes into APC mode as an antigen-presenting cell. It does what with that antigen? It processes it, so it basically degrades it with all those enzymes that tear it apart, and then it expresses it on its surface through MHC class 2, right? So MHC class 2, you have these peptides that are presented and ready to be shared with T helper cells in those lymph nodes. And this is an important interaction that's happening there in the lymph node, this interaction between the B cell that is presenting this antigen and the T cell. There's going to be a lot of signaling both ways. B cell is saying, this is what I found, this is what's presented to me, and that T cell has to basically lock in and say, yep, confirmed, and it starts giving cytokine signaling stimuli, basically, that will tell that B cell, go ahead and be activated. This is like the confirmed thing. That's really important in the adaptive immunity, that you have all these confirmations, checks, validations, etc. So you get that stimulation, the B cell is now fully activated, its target has been validated, it is locked on, and it goes through what we would call affinity maturation, which means that B cell receptor, as a cell population starts expanding and proliferating, it plays around with that B cell receptor even more with those new cells that are dividing, and it will try and find like minute, small differences that improve the the affinity to the antigen even more. That's affinity maturation. So it's like the very best of the best binders are selected for at the end of that affinity maturation process. And those end up being the B cells that become plasma cells, secreting antibodies, or memory cells. But for this T cell help, again, in this third panel, one that's level 3, that's why it's so critical for that T cells. It's almost like it gives it encouragement. So it sends its signals in the form of cytokines to help it mature and be activated. It has that CD40 ligand on the surface of this T cells that are important also for that warm, fuzzy handshake. All of those things are important to help make sure those B cells are fully activated. Okay, so we have our fully activated B cells. Most of them are going to become plasma cells, which means they're no longer, they're primarily going to be secreting antibodies out from them. On a genetic level, some expression will take place that basically removes the transmembrane domain. So when they start expressing those antibodies, it doesn't any longer get targeted up to this plasma membrane. It gets secreted out of the cell altogether. So the secreted form, this is basically what it looks like. We'll talk about a couple other variations in this form. But when we look at this molecule, a couple features. So one, this is a monomer of an antibody, also known as an immunoglobulin. What does a monomer mean? Just one unit. What would a dimer be? 2 units. And a pentamer? 5. Okay. So this is a monomeric form of a single antibody, a single immunoglobulin. And the way these things work is you have your antigen binding sites, which are at the very end of those Y-shaped molecules. And those antigen binding sites on these antibodies will have 2 different chains that are responsible for touching and interacting with that antigen. One of those is a heavy chain, so think of it on the being on the inside of your arm, and the other one is a light chain. That heavy chain, if you follow it down in this cartoon, goes all the way down to the stem or the stalk of that antibody at the feet, right? But the light chain terminates basically, I guess this would be analogous to the armpit, right? It terminates. So you don't have as much light chain on that antibody as you have heavy chain. These are historical terms if you're wondering how they got those names. It refers to the mass of, the relative mass of these protein subunits. Heavy chain is about 2 to 3 times heavier than light chain if you were to separate out by ultracentrifugation or electrophoresis. So don't worry about that, but just know that the heavy chain is the one that's kind of extending through the entire length of that antibody, and the light chain is only going as far as like that armpit level. So let's put that armpit level in more technical terms. We have what's called the Fab, or fragment antibody, and that is like the part that's specific for recognizing whatever different antigen it encounters. So fragment antibody, Fab, the arm region, and then we have the FC region, which is the stalk or the stem of the antibody. It stands for fragment crystallizable, which is another historical term. Any guesses what the FC region does on an antibody? Some process.
Whatever does antigen
Kind of. Any,
Is that what's on the B cell? Like reports for this?
yeah, say again.
Signals for the phagocytosis macrophages.
It could, yeah, so you said it could be a signal for phagocytosis for some phagocyte, right? So it could be a signal. But basically, generally speaking, it has the effector function of that antibody. So it could be a signal for a macrophage to come eat me, or it could be some other signaling thing. But the FC region, people tend to write this off as not very important, but it determines what the antibody is going to do, what mechanism it uses to clear whatever antigen is on its Fab region. So we'll talk about that a little bit as well. But FC region is very, very important.
An analogy, help remember that, be like it's a tuning fork. Sets off certain waves. He's done.
I have an analogy for you. Yeah, maybe it's maybe tuning forward, we'll circle back to that, antibodies more. Okay, so the Fab region, let's say I am at a firing range and I've got a target, right? I've got a target down range, let's say it's an easy target, it's like 100 meters away. Got my target, but I've also got like 10 million other targets around, right? I've got, my firearm is a pistol. Yeah, I know.
Easy shot. Easy shot.
More than it. Let's say I'm not super happy with that pistol, but I know I want to keep hitting my target, and you know I have got my only my target obvious million that I want to aim at. So the FC region is more like which type of weapon I'm using to destroy that target. So I'm gonna chuck my pistol. I'm gonna pick up like a grenade launcher, a shotgun, a rifle. I don't know, like something bigger. But point is, like this means that you are just locked in on that one target. But the FC region is sort of like the effector, it's what's effectuating the mechanism of destruction. Does that work, or is that the most ridiculous thing that you've ever heard?
You did.
Don't answer that. Made sense in my head. Okay, fine. Tuning fork it is. Okay, so antibody isotypes. This is about those FC regions and kind of like an indication of form pulse function as we've been talking about. But these different classes of antibodies that perhaps you've heard about before, like IgM and IgG. What's not different about them is their specificity for a specific type of antigen, right? The Fab is the same, or could be. But what's different is they all have slightly different FC regions, and those FC regions will dictate how they handle that pathogen or antigen. Okay. So IgG is probably the most well-known of the immunoglobulins, of the antibodies, this particular isotype. It is one of several isotypes that exists exclusively in a monomeric form. It doesn't complex with other IgGs, just secreted off on its own as a little monomer, and so is IgD, and so is IgE. This is the one that is most abundant in your serum. It's longer lived as far as proteins go, it has a longer half-life, and because it's so small, it is able to do a lot of things that some of these bigger forms of secreted antibodies can't do. So for example, IgG can cross the placental barrier. How might that be useful for, let's say, a fetus?
Like an RH negative mother, like RH positive fetus.
That's with regard to blood type, but I'm thinking more strictly along the lines of offering protection to the baby. Yeah, so that fetus is not going to have much of its own immune system, right? Its own B cells are just starting to form, so it needs maternal antibodies. Well, it's still in utero before it's even born. It needs some of those maternal antibodies. The ones that can do it are these guys, these light little IgG molecules that can cross the placental barrier. A big guy like IgM, probably not going to do that. So IgG, most abundant in serum. It is good for opsonization. Ooh, we'll come back to that in a second. Yes, ma'am.
For those like antibodies that transfer to the fetus, will they stay like forever inside the fetus? Or
No, they don't. That's a great question. So, when the baby is born, it will have in its own system maternal antibodies that persist months, possibly up to maybe 2 years. But those, as soon as the baby is born and is no longer actively receiving IgG across the placental barrier, those antibody levels start to wane, meaning they decrease. So that's why 2-year-olds are so susceptible to diseases, right? Their maternal antibody levels are starting to decrease, those titers are dropping off, and they're on their own to produce their own immune defenses.
so those IgG, they won't be produced at all inside the fetus?
It's just a protein, it's not a self-reproducing molecule. So once IgG is secreted from that plasma cell, it has its own duration, its own half-life, like any protein does, So yeah, it will probably get degraded over time. It will not last the life of that child who receives it, that new person. Yeah. So. Yeah, so IgG, you've got opsonization, which we talked about earlier. What other molecules did we talk about that is really important as an opson?
Complementary.
Complement, yes, exactly. So in your own words, and we'll see this in a couple slides reiterated, but what is opsonization?
Throwing a target on the enemy so that we can easily identify.
I like it, yeah.
Makes it more digestible.
Yeah, it's the coating of some target with an opsonin, like complement or like antibody, to make it more attractive or appealing for things like phagocytes, things that will be targeting it for removal. So IgG, for example, has an important role to coat things like bacteria and make it more attractive for phagocytes that are coming around. It's an important role. ADCC, that's antibody-dependent cell-mediated cytotoxicity. We'll talk about that in a couple slides. So yeah, IgG is arguably one of the most important one of these isotypes. However, it's not the first one that is produced once a B cell is activated. The first one that gets actually produced and secreted is this big guy, the IgM pentamer. And the reason why IgM is so good as a first responder is that it has multiple different antigen binding sites. So the more you throw together, in theory, the more effective it can be at very quickly starting to tie up and aggregate targets together. That's principally how IgM works. It aggregates pathogens together. Because it's so big, it's not very good in tight spaces like tissues. It doesn't work very well there. It operates better in the bloodstream. So when you get like the initial bloodstream infection or something like that, IgM is very, very good and it's a rapid responder to bind to some of those targets. With IgA, we'll see that in a couple slides too. This is the one that's kind of associated with mucosal surfaces. You'll oftentimes see an S before that, like S-IgA for secretory IgA. It's secreted in those mucosal surfaces. And part of its job is just making sure that you can neutralize some of those pathogens that are commonly encountered in those barrier regions, like your mouth, like your GI tract, etc. IgE, this is an antibody that has been specially evolved to handle parasites. So it has a more intimate relationship with mast cells and eosinophils, those innate cells that are involved in parasitic response. And then finally, IgD is not a very well understood isotype. It is not secreted. It remains on that membrane-bound form of B cells. And we probably won't talk about IgD again. Yes, sir, I'll come back to you in just a second.
How is, because the IgM is so big in structure size, how is it expressed on a B cell? Like, what does it look like?
Yeah, that's a good question. So, like, when a B cell is activated, when it still just has nothing but BCR on its surface, you will have an activation of genes that start encoding for IgM. IgM basically will self-assemble into pentamers while it's still inside the B cell after it's been translated into protein. It'll go to the endoplasmic reticulum, it'll get modified, it'll go to the Golgi, and then the Golgi will package that intact pentameric structure into vesicles, which get trafficked to the surface and released or secreted. So they don't stay on the surface, they're not membrane bound, they are secreted from the B cell. Do you have a question too?
Yes, ma'am. So I know in this example, like the pentamer is made up, looks like of IgE isotypes. Is it always that way, or could it have like 5 IgDs attached to it? Is that a thing, or not attached to it, but composed of 5? Does that make sense?
I think I get what you're saying.
Yeah.
So this cartoon, it might look like this has stock from another isotype, but it
Yeah.
doesn't.
Okay.
This is all IgM, and there's a dedicated genetic cassette that corresponds to IgM.
Okay.
And you'll see that in another slide. Yes, sir.
And for IgM, it having so many binding sites, does it have different VABs for different antibodies to?
No, because it originated from that one B cell, and that B cell in its genome, it has already undergone somatic aggregation and commitment and recombination to only expressing that one BCR.
So you just pick up more of whatever that specific target is.
Exactly, yeah. So going back to just for a moment, like this slide here. This is, let's say, expressed that BCR is like IgD or IgM, it's membrane bound, it's antibody. But this particular antibody, this B cell is through and through specific for that one antigen. So when it gets activated, the secreted antibodies are only gonna be for, like, all those antigen binding sites on those antibodies are always gonna be specific for that one antigen. With affinity maturation, the confusing thing is that there's a little bit of additional tweaking of those antigen binding sites, but it's still essentially a clonal population, so it's not gonna see something different. It's gonna see it just a little bit better, that's all. Yes?
So just for that example, it would be like, the cell is hard-coded on strep antigen. It encounters, the strep antigen, and all the IgM that it produces is going to be anti-strep.
Exactly. Not just anti-strep, but like exquisitely specific to one aspect, one tiny piece of that streptococcus pathogens. So yes, exactly right, yeah. It's gonna be blind to other things. Okay, so, that kind of brings us to the topic of class switching. So this is why I liked my stupid analogy about being on a target range and needing a different weapon sometimes, and needing to upgrade. So you want to retain your target, right? You're retaining that, but you need to change effector function because you're moving along and you're progressing in this response. When IgM is the first on the scene, it has a specific function, and it can only do so much. It has limitations. So that specific B cell that is specific for some streptococcal antigen, when it has done all that it can do as an IgM, it will undergo what's called class switching, which means on a genetic level, some of these earlier genes that would allow it to become IgM or IgD are actually cut out through a mechanism called mRNA splicing. And what is left available at that point is only the ability to become IgG, IgE, or IgA. And there's a logic or a rationale to the order of these things. You need IgG in the most abundant, so that's the next closest to it. So what will happen is you will start producing increased amounts of IgG, but the Fab region doesn't change. You still have that exquisite specificity. The only thing that changes is the FC region. So that's what class switching is. Same recognition, but different effector function. So we're going from an IgM, which can happen quickly, like within, let's say you get that streptococcal exposure on a Monday, within 4 days you had activation and expression of IgM out into the serum. Okay, so this is pretty rapid. It takes longer for class switching and the arrival of IgG because you need to undergo things like affinity maturation. So it's a little bit longer. But as soon as IgG arrives on the scene, it's usually a more specific molecule to handle those antigens, and it's able to traverse more tissue types because it's smaller. So beyond that, you can do class switch to other isotypes as well. You can class switch to IgE. If you have signals that indicate you're dealing with a parasite, then those B cells will be stimulated to class switch to IgE molecules. Or if you need stimulatory response, then class switch to an IgA.
I think it was depicted on the picture, but that mRNA splicing is all happening inside the B cell, kind of like upstream before they are releasing. So like for a B cell that hasn't become a plasma cell, is that?
No, it has. Usually, so like a plasma cell is secreting the IgM, right? It's secreting it as a plasma cell. It's going to receive some signaling, probably in the form of a cytokine. It says we need class switching to start happening. We need you to be an IgG now. We need you to start secreting IgG. So it receives signaling, which does translate to usually transcription factors and other processes that ultimately result in the increased expression of these genes that are IgG genes. So you can make IgG molecules.
Okay, that's happening in the B cell.
That's happening in the B cell. The B cell receives those signals from cytokines and it responds. Yeah, it responds by making those IgG molecules. Yeah.
And to circle back, I think there was like 2 or 3 slides back, that was with the interaction between the B cell and the helper T cells, is that?
In
part of
part,
that, or-
yeah, in part, but not exclusively too. So that T helper cell interaction with the B cell is going to be really important for signaling and communication. Some of the signals that the B cell is receiving from the T cell are in the form of cytokines that are pro-activation, pro-proliferation cytokines. But that's like the initial activation. Downstream, after they've separated, that B cell will continue to receive cytokine activation signals. It has the cytokine receptors on its surface, and those receptors are receiving constantly communications from other cells saying, we need you to be this, we need you to be that. Okay.
Ma'am, so for the class switching, can the plasma cell itself switch classes, or does it need to go back to the B cell and have to produce a new set of plasma cells?
The plasma cell itself can, but mRNA splicing is an irreparable thing. So it becomes shorter and shorter in its one ability, like that one plasma cell's ability to class switch. So like you can, like the scissors here, I don't know if this is very clear, but when a portion of that cassette is spliced out, that's a permanent thing.
So you couldn't go from N to G and then go back to D because it was
No,
cut out?
you splice that out. Yeah, but you can trek along and become IgG, you can become IgE, IgA, but yeah, you can't go back to those spliced out regions. Was there another handout? Oh, yes, ma'am.
Is it for the class switching, is it only from IgM to different classes, or the other classes can interchange within them?
Yeah, that's a good question. So you do always start out as IgM, like that B cell early on in its life, right after it's been activated and it becomes a plasma cell, it does start with IgM. And then from there, it's more a combination of different signals and cues it receives. But yeah, starting place is always IgM. Okie doke. All right, and then at this point, this slide should be like second nature to us, but just to kind of foot stomp some definitional terms. An antigen, which you'll see in my shorthand, probably the duration of this course, I see it abbreviated to AG. This is what is actually recognized by either the specific antibodies, the abbreviation for antibodies, AB, or the T cell receptors. Okay. On this antigen, so let's say this is an antigen from some microbe that your immune system encounters. The part that the antibody is actually recognizing is called an epitope. So here you can see some arrows on one big protein antigen. The point here is that antigen is like a big molecule, and those antibodies recognize only 18 to 30 amino acids typically if it's a protein antigen, small amount. So you can have multiple antibodies recognizing to multiple different epitopes on that protein antigen. This term down here, paratope, you don't hear it as often, but the paratope would be the part of your antibody, that antibody itself that's recognizing and binding to the epitope on the antigen. So not very common, not as useful, but epitope you'll hear a lot, so you need to know what an epitope is. And then there's a subtle distinction here. Sometimes you hear the word immunogen used interchangeably with the word antigen, but the subtle distinction is that an antigen is something that can be recognized by your immune system. An immunogen is something that actually triggers an immune response. Most of the time they are the same thing, but not all antigens actually successfully trigger an immune response. So that's a subtle difference there. A bit of a nuance. And then in terms of what actually works as an immunogen, not all classes of macromolecules are equal in the eyes of antibodies. Some are better than others. The best and the easiest in terms of forming a very tight bond, high affinity binding, is a protein target. So there are a lot of different, like let's use a bacterial example, bacterium that has a long flagella. That flagella we've already talked about, it's made up of what? Protein, flagellin specifically, and that is like candy to immune cells. They love protein, and they form, that's super immunogenic. So Your immune system recognizes it and it forms really good antibodies against that flagella. And capsule doesn't cover flagella, so it's not protected if it has a flagellum like that, it's like exposed. A little bit worse are carbohydrates, but we have to be able to deal with carbohydrates. We have to be able to see them because there's so much sugar out there. What is capsule? We talked about that covering on bacteria. What is that?
Bunch of carbs.
Defense mechanism.
It is a defense mechanism, but like what's it composed of?
Carbs.
Sugars. Yeah. So in order for our antibodies to have a ghost of a chance of clearing all these pathogens, and there are a lot that are encapsulated, you have to be able to recognize carbohydrates. And that includes viruses too. Viruses have receptors, which are called glycoproteins, and that name tells you what? Sugar-coated proteins, basically. Yeah. So the glycosylation on some of these proteins is extensive. So that's a really important thing. It's not as good of an immunogen, but it's important that we can recognize it. And then down on the bottom tranches of recognition, we have lipids and nucleic acids. They're just poorly immunogenic. We can still recognize them. Our antibodies are able to identify nucleic acids. They can bind to them. They can even bind to lipids, but they do not stimulate a very robust immune response. Some more terms to be aware of: monoclonal antibodies versus polyclonal antibodies, MABs, sometimes known as MABs. Anybody ever heard of monoclonal antibodies before? Yeah, hopefully a lot of you guys. In what context have we, or would we talk about monoclonal antibodies? Sort of, close. Thinking more of like a passive therapy. When you give somebody a monoclonal antibody, yeah, go ahead.
Was it during COVID? I think it was in Florida. They were giving people
Yeah, quite possibly. There were a number of monoclonal antibody countermeasures that were produced during COVID. But monoclonal antibodies were basically an artificial lab-generated medical countermeasure that takes advantage of the specificity of the Fab region of antibodies. So you can design and engineer a Fab that is exactly targeting whatever you want it to be, and mass produce it in these immortalized cells called hybridomas, and then clean it up and put it out, through the pharmaceutical pipeline. So we have a bunch of different monoclonal products, monoclonal antibodies, but know that your body does not make such a thing. You make polyclonal antibodies. So, and the reason why you make polyclonal antibodies is because of this, because when your immune system sees like a bacterial cell or a virus or a parasite or whatever, it's going to generate antibodies to all different parts. So these are polyclonal. They're not recognizing the same exact thing. They're recognizing different components of that antigen. In terms of affinity versus avidity, you've probably heard me say affinity once or twice. What affinity is it's a technical term that can be used to quantify the intensity of this binding interaction. It can be applied to a bunch of different things, but when we're talking about antigen-antibody binding, high affinity binding is what you're going for. You want to have high affinity binding. But you usually don't get there until you've done some affinity maturation. So you start off relatively weak with low affinity, and then after affinity maturation, you've fine-tuned that binding interaction, and you come up with an ultra-strong binding capability. You also, as this little diagram is illustrating, you have two arms, assuming you're dealing with a monomer antibody, you have two-armed molecule. So if only one of your arms is tied up with an antigen, you don't have the same strength of ion interaction as if you had both of your arms with two antigens. So the KEQ, that stands for, it's an association constant that gives you an indication of the strength of this interaction and makes it quantifiable. So if you have both of these antigen binding sites tied up, you have a much higher association constant and a much tighter bond. With avidity, this refers to the overall strength of a multivalent binding between antibody and antigen. So this could be considered the start of a high avidity response where you have both of these antigen binding sites taken up as opposed to one. But where you really see this impressively displayed is when you're talking about like a pentamer of IgM. So how many binding sites would you have on a pentamer of IgM? You'd have 10, right? So you have 5 molecules with 2 binding antigen binding sites per 10. If you add that all together, you get like a super high avidity capability. Okay, so the next couple slides talk about these effector functions in a little bit more detail. Oh, actually, yeah, let's take a 10-minute break. This is good stuff. Before you go on break, just be aware of my horrible typo there. This should be and. So, it's 13:44. Come back at 13:54.
I'm back at it. Charles Brown actions. But yeah,
Exactly. Yeah, and there's a different strategy.
a lot of outside.
The nice thing about that is the true you don't get as robust responses.
Read them, just read the slide. You know, same quality.
Yeah. I would like some examples. Yeah, so what I did. COVID, I mean, that's like the first. Yeah, I don't know. I was.
Yo, so like like your skill goes with the trip. So it's like after you talk there, and then you get a reference to it. Now, go to Outlook and then type in Unity. And I'll take Unity page.
Sweet. That's
Big micro. No idea what's going on.
I feel like that for people like if that's something you don't think is different way to get the test and it shows a different way, then you might not use it.
No, actually, it's helped me. That's how I feel.
Yeah, it's a lot worse.
I swear, I see your face, because I make yeah. This easy shit. Yeah. But if you, I don't know take that with the brain.
Hey, that question is my least favorite.
So you just run the test account, check out your
In terms of being able to detect their markets, microscopic is not possible. So usually how we characterize these is
It's a lot working. I'm so glad. I'm used to
So there's a couple different types of transmission. And both of them are, and then there's other ways that you can indirectly get structure and things like that. You can actually indirectly reconstruct something based on like direct recognize yeah.
Because I'm curious, like, is it actually those shapes or is it so small, right?
Yeah. No, no worries. I wouldn't want to listen to either. No, just kidding. Okay, so we are making good progress. I think we'll get done today. With these effective functions, we'll start off with. We can go through these pretty quickly because they're intuitive. They're not super difficult to understand. And there's just a couple main categories that you can get through. I have pictures on all these series of slides about the effective functions of antibodies. Fortunately, we have a common theme to carry us through. The examples in all of these cartoons is how antibodies will control this virus called SARS-CoV-2, which we're all too familiar with. Okay, so the first function is arguably the most important, certainly the most well understood. People, you'll hear this, if you haven't been hearing it already in your careers, you'll hear it as PA. You're talking about neutralizing antibodies. You're talking about antibodies that can bind pathogens and toxins, physically blocking their ability to infect cells or cause damage. Pretty intuitive stuff. These are kind of some subcategories on the mechanisms by which neutralization can occur. Most of these are pretty intuitive, but this first one, for example, is maybe a little bit less intuitive. Steric hindrance. What does that mean? Yeah, exactly. Yep. So if you're like, let's say you're in a hallway, have you ever been in a pretty good sized hallway and somebody's taking up maximal space where you can't get by them? You know, that's like an example of steric hindrance. Or in that narrow door frame right there, somebody's just blocking it with their body. That is the same, antibodies can do the same thing. They can prevent, pathogens or toxins from doing their thing because of steric inputs. They can also do what's called receptor blocking, which is what is shown in this cartoon here. We've seen this cartoon of SARS-CoV-2 virus particles before. Those spike proteins are glycoproteins on the outside of the of the virus. And those glycoproteins are responsible for interacting with the cognate receptor on the host cells. So in this case, it would be the ACE2 receptor, which is expressed in abundance on cells like respiratory epithelial cells, right? If you have antibodies that are specific for the glycoprotein of SARS-CoV-2, what those antibodies can do is prevent the binding interaction between glycoprotein and the ACE2 receptor. If you prevent that from happening, no entry into the cell, no infection, no problem, okay? So that's how that works. You can also have toxin neutralization. What kinds of pathogens are we worried about for toxin secretion? That would be, are you thinking of endotoxin? Okay, and no, that's perfectly fine. What about exotoxins? Anybody think of really famous exotoxins? Say again? No, that's the same thing as here. That's an intrinsic endotoxin. Botulism toxin, yeah, Botox, yeah, for sure. Clostridium botulinum secretes Botox, botulism toxin. What about anthrax toxin? Anybody ever heard of that? What about pertussis toxin? Cholera toxin? A lot of different bacteria secrete toxins. Doesn't even have to be bacterial. It could be some parasites have toxic components that they shed away. And it could be toxins from even animal bites, for example. We're talking about snakes and their snake venom has toxin in it. That's exactly what antivenom is. It is antibody that blocks the toxins in that snake venom. And finally, we have agglutination. What does that sound like? Yeah, aggregation, clumping together. So when these antibodies start recognizing antigenic targets, they can cause them to clump together. And imagine if something is like just hanging all over you and you're trying to move around. You can't do that anymore because you're aggregated together. So if you do have a bunch of neutralizing antibodies, that is great. It'll either prevent the infection altogether in some cases, or at least reduce the severity of the infection. Very powerful way of combating toxins and pathogens. Just out of curiosity, is there a duplicate slide for neutralization in your deck? Okay, sweet. Okay, and then another important effective function is opsonization, which we know very well now. So in this example here, we have a viral infected monocyte. So SARS-CoV-2 has gotten into this respiratory cell, it's infected it, and since this is a nucleated cell, it has MHC class I on its surface. So it's basically, as it's dying, it is communicating the fact that it is infected with a specific virus by virtue of its presentation of this antigen on its MHC class I. This phagocyte will recognize the antibodies that have bound to that presented antigen on MHC class I, and it will make it that much more attractive to that phagocyte, that macrophage, whatever it is, because it has a receptor, this Fc receptor on those macrophages. Remember that cartoon of the multifunctional macrophage? Macrophages and other phagocytes will have receptors for the Fc region of antibodies. So let's see, opsonization can also trigger antibody-dependent cellular phagocytosis. So, yeah. The third major effector function is complement-dependent cytotoxicity, or antibody-mediated cell lysis. We talked about complement a little bit and what it does. One of those mechanisms that we talked about was the formation of the membrane attack complex, the MAC complex, which could punch holes in bacterial cells, or in this case, virus-infected host cell with SARS-CoV-2. So the way this works is almost identical to what we just talked about with opsonization, but when you have the antibodies that are recognizing antigen that's expressed on MHC class 1 of these affected cells, instead of a macrophage coming along to phagocytize that cell, or some other phagocyte, instead you have the complement molecules, complement proteins coming along. Complement proteins will recognize the Fc region of those antibodies, and they will activate the complement cascade. When the complement cascade is activated, one of the outcomes could be that membrane attack complex. So ultimately what will happen is this cell will be destroyed by a mechanism that we haven't really talked about much, but when it has that pore that was formed in it, it can no longer maintain integrity or osmolarity, and it just bursts, it lyses. And finally, the fourth method, the fourth effective function of antibodies is antibody-dependent cell-mediated cytotoxicity, or ADCC. Very similar story as the other ones too. Presentation of antigen on the surface of an infected cell, and antibodies recognize that, and then instead of a macrophage or a phagocyte, you have some cytotoxic cell. It could be a natural killer cell, or it could be a CD8 cytotoxic T cell. Either way, they recognize it, they have receptors, and they initiate apoptosis in the infected cell by releasing enzymes into the target cell to kill it. Okay, getting a little bit more into immunological memory and how that works and tying that in with vaccines, we'll talk a little bit about memory B cells, memory T cells, and how long-term protection works. When we're talking about immunological memory, this little graphic at the bottom right is sort of helpful in helping us pull everything together in terms of like our primary immune response and how that transitioned from primary response to a much better secondary response. In this example, this is showing a vaccination, not a natural infection, but this is similar to what happens in a natural infection as well. But if you've ever wondered why you get a booster when you have a vaccination or why that's sometimes offered, this is why. So what happens when you get an initial vaccine, it's, let's say it's your first time that your body's ever seen this particular antigen. Let's keep with the COVID example, because prior to 2019, none of us have ever been exposed to that virus, right? So none of us had ever mounted an immune response to that particular type of coronavirus. And this was kind of a novel thing when we started getting vaccinated, assuming we hadn't already been naturally infected. So we would get our initial exposure to SARS-CoV-2, and within a couple days, those naive B cells that were hanging out in your lymph nodes would receive that antigen. It would be presented to them in those lymph nodes. So those B cells would become activated, they would become, the specific ones for SARS-CoV-2 would become activated, and they would get signals to proliferate. And they would proliferate a fair amount, but not a huge amount. And they would become either plasma cells or memory cells. For a short time, they would be secreting antibodies. But they're short-lived cells, relatively speaking. So most of the time, most of those plasma cells are going to be destroyed after the infection. The infection is cleared, and the majority of those plasma cells stop being produced. The small amount that persists as memory cells will linger on for a much longer period of time. So let's say 3 or 4 weeks later after that initial vaccination, you go back for your second booster shot. At that time, those memory cells are going to be confronted with that same antigen that they saw initially with the primary vaccination. Only this time the immune response will be much, much different. It's going to be faster. Look at the kinetics of this response as these memory B cells become plasma cells and secreting antibodies down. It is steep because they know what they're looking for and they can be very quickly stimulated to differentiate. So much, much faster. Instead of a week, we're talking about days before they start pumping out antibodies. They also have that firm handshake, all those co-stimulatory signals and cytokines being released much faster. So they get that warm, fuzzy feeling much faster and they start producing even more antibodies. So more antibodies, the quality of the antibody antibodies is better because they've undergone affinity maturation, so you have super high affinity binding, much faster timeline, and higher secretion of those cells. From that point on, after you've had that secondary immune response, you usually will have higher levels of IgG persisting in your system at that point. And you will also have some specialized plasma cells that we haven't talked about called long-lived plasma cells, as opposed to the short-lived plasma cells. These guys can live for a significantly long time, not as long as memory B cells, but the special thing about them is that they continue to secrete antibodies. The memory B cells do not. They basically do nothing. They do absolutely nothing unless they're confronted with that antigen again down the road. So let's say you get your third exposure to COVID, memory B cells will kick in at that point. But meanwhile, those long-lived plasma cells will be continually expressing antibody in the form of IgG. So those are some of the main differences. Primary immune response is fundamentally different than that secondary immune response because it's doing something that is much more difficult. It's always harder to do something if it's your first go at it, right? You've got some work to do. So it takes much longer. You don't get the same amplitude or magnitude of antibody expression in those plasma cells. You don't have that nice high affinity binding that is occurring to those antigens. The T cells may be naive as well, relatively short-lived. The first antibodies are produced are those kind of crappy IgM tumors that don't have high affinity binding. And because of this, even though you have an antibody response, that person, depending on the pathogen that they're being confronted with, could get sick, right? So the goal is by the time you get a booster shot, you're pretty much making that immune response so fast that the person doesn't get sick at all. But how many of you guys remember when you got your first COVID shot, what that felt like? Sucked. Yeah, that was a pretty brutal response. I was in ILE, I was down in Fort Leavenworth at the time, and we had about 850 field grade officers in our cohort. We all got vaccinated in the same 2-day window. And they had to cancel classes the next day because everybody was just sucking. I mean, it was a really robust immune response, and it was miserable.
For me, it was the second shot.
It did it?
Yeah. It
That's
wasn't, I was fine after the first one, the second one is what put me down.
Did you receive, was it the same vaccine in both shots? Do you remember?
That's a good question. I just mixed it. They just told me I needed to take it.
Fair,
Yeah.
yeah. Do you remember?
Yeah, it was the same shot.
Same
Or
shot?
the same, I think it was
Same.
but yeah, the second time was worse.
Okay. Interesting. I mean, mine was definitely worse the first time because I was afraid to get the second shot.
Question for you, so,
Yeah.
when I got my booster, it was long ago, but I remember in the axillary, like, lymph node was swollen, like, there was a ball.
Yeah.
Is that just that rapid proliferation of memory cells, like the cells, like, because that response to the second one? Is that
Yeah.
what it is? What was it?
Exactly. So, wait, did you get Moderna or?
I think with Moderna and Pfizer, I had to stop at the second.
Okay. I guess it doesn't really matter which vaccine you got, but like.
Or no, I didn't, I'm sorry, it was the third that I got different ones, they're both Moderna.
Okay, so those, like Moderna, for example, that's an mRNA vaccine, and we'll talk about that in a second too. But when you get that initial shot, what is happening is that mRNA is going into cells and they start expressing the protein that is encoded by that messenger RNA, which is spike protein. That's the most immunogenic part of this. So your body is producing, ultimately the goal is to produce a lot of antibodies against spike protein, right? But in the process, it's active immunization, so your immune system has to do all the legwork. And that entails like some pain in the process. You get that initial expression of this foreign molecule, spike protein, and your immune system is doing what it does, and immediately, like the cells that are producing it, they're presenting it to antigen-presenting cells. The antigen-presenting cells, say you got it in your arm, which you probably did, right? Yeah. So those antigen-presenting cells are going to migrate to the Varus lymph node, which is where probably.
What was axillary?
It's axillary probably, right? So that's a major lymph node, high concentration of lymph nodes in the axillary area. And that's where a lot of that, like, rampant communication and kind of like a freak out. Lots of excited immune cells gathering together and saying like, we've encountered something that's legit, we need to form a response to proliferation of all these immune cells, and the lymph nodes themselves actually increase in size, lymphadenopathy. So yeah, I mean, that's exactly what happens when you have a robust immune response. And that's usually why it happens in the armpits if you get a shot in the arm. Yeah. Okay, so I don't think this diagram really tells you a whole lot that we haven't already talked about, but just a little bit more detail on memory B cells. I'm gonna walk the dog through this entire process here. So we have, let's say, spike antigen that your epithelial cells and other tissue cells are starting to produce. That spike protein is recognized as antigen by these antigen-presenting cells. It's presented to B cells. Certain B cells that have specific B cell receptors for that spike protein will be activated. They will be activated to proliferate, and they will be activated to become plasma cells. Plasma cells will be producing those antibodies, which will start off as class what? Which isotype? IgM. Will start producing IgM molecules, and then they will class switch and start producing IgG antibodies against spike protein. But regardless, those antibodies do not last forever, so those memory B cells that persist should be stimulated to persist and continue on for a long time. So when you get that secondary response again, second confrontation or second exposure to that spike protein, immunogen, boom, you get increase in plasma cells and you get additional memory cells as well on a much faster timeline. With those memory B cells, these characteristics, it is long-lived. It can be for the rest of your life, possibly, if you're lucky, or at least for years and decades. It depends on the specific vaccine and it depends on the pathogen. Some of them don't have that long of a duration. But you should have very high affinity surface antibodies. You should have rapid activation. And these are pretty much the same things we saw on the other slide. You have higher quality antibodies being produced and more of them, and the ability to differentiate into plasma cells as memory cells. Okay, so that should set us up now to have this short conversation about vaccination a little bit. I just animated this, so you'll see the rest of your slide in a second, but it's a very busy slide. For vaccination, what we're trying to do is mimic the what happens when you have a natural infection, but do it in a controlled way so you don't have a significant pathology. So just kicking in to induce an immune response. A well-designed vaccine is going to initiate a robust immune response that will last a long time. Just like when we talked about the difference between, like the semantic difference between the word antigen and immunogen, there is another semantic difference between the words vaccination and immunization. It's also a bit nuanced, but it's worth knowing. So you can vaccinate someone, and if for some reason they fail to mount an immune response, they can be vaccinated but not immunized. Immunized means they've actually mounted a successful immune response. So there's a subtle difference. Most of the time, you can use them interchangeably. But going back to my example from last week when I was talking about hepatitis B vaccines, those are subunit vaccines. They're not the greatest at stimulating an immune response. So like when I said I've been vaccinated several times and I don't have antibodies to Hep B, not immunized, but am vaccinated. So that does happen, but most of the time you do stimulate a good immune response and you get immunized. In terms of the difference between natural immunity versus artificial immunity, with natural immunity, mostly we're talking about you getting an actual infection from a real pathogen that you encounter all the time. Even there are a couple notes in other terms to know. An active natural infection is one where you just encounter a pathogen naturally and you raise an immune response all by yourself. However, there's another type of natural immunity that occurs, natural being not for medical, not countermeasures, and that's passive natural immunity. This is when you transfer antibodies from one individual to another. So you didn't make the antibodies, someone else did, and this is happening naturally. How on earth would that happen if you're not talking about like blood transfusions or pills or something? With the fetus, exactly. So mother to fetus in utero, you have antibodies like IgG that can cross the placental barrier. That's an example of passive natural immunity. It's been going on for a long, long time. It works really well for that fetus. Then, when the baby is born, they can get passive antibodies that they didn't make themselves, their mom made them through drinking breast milk. So that's another example of passive natural immunity. When you're talking about artificial immunity, this is something we've only developed in the last century with any kind of efficacy or proficiency. We've been vaccinated for even longer than that, but artificial immunity is usually associated with vaccinating people. So whatever type of vaccine you're using, you're stimulating that immune response. When you have a passive artificial immunity, that means that you are giving that person antibodies that they did not make themselves through an artificial way. And that would be like my example that I used earlier, where you have a snake bite and you're giving antivenom, or you have somebody who has an infectious disease process, but it's like perhaps a toxin-producing bacteria. So like the Tdap vaccine, you guys heard of Tdap vaccine? What is that vaccine?
Tetanus, diphtheria,
Tetanus, diphtheria, pertussis. So the tetanus part and the diphtheria part of that tripart vaccine are both targeting toxins. They're targeting the diphtheria toxin and they're targeting the tetanus toxin. The pertussis part is an acellular subunit part. The other two are against the toxin. So, and also, if you have somebody, another example I can think of. Has anybody ever gotten bit by an animal and had to receive rabies post-exposure prophylaxis? I've done that before.
I've given it, I've received it.
Okay. So
I didn't get rabies.
So there is a vaccine for rabies. You go through that entire active artificial vaccination process. But if you have somebody who's in an emergency situation and they just got a bite from a rabid animal, from an animal they think is rabid, rabies is a usually fatal and fast-acting viral disease. So it's really, really important to protect them as quickly as possible. Because of that, we have HRIG, human rabies immunoglobulin, which is an immediate medical countermeasure, and it's basically just an antibody against rabies virus that you can give that person. So you circumvent all of the natural steps that have to occur for that person to raise their own antibodies, and you go straight to just giving them antibodies against rabies virus. There are a couple other examples too of just giving somebody an antibody treatment to skip over the steps, but that's an example of passive artificial immunity. Okay, so we talked about how this works. You're generating those memory cells, both a humoral response from B cells as well as a cell-mediated response from T cells. You get those memory cells, and those memory cells are quick to be reactivated if you get exposed again. The benefits of vaccination, hopefully everyone in this room is sort of like a believer that vaccination is an overall good thing, hopefully. But let's enumerate some of these benefits a little bit. Obviously, the clearest one is that you yourself are protected from disease, individual protection, and that's a nice thing. More importantly, on a population level is this concept of herd immunity. You guys know what herd immunity is? Yeah, what is it?
Enough people in a room are vaccinated, that the virus will spread to aren't vaccinated, and they get all protected.
Yeah, exactly. Okay, so it's meant to vaccinate or protect, immunize such a large percentage of a population that it makes transmission much less likely. So you get like a population level immunity. When we get to block 4 and we talk about measles, you'll hear me on my little soapbox. What's happened recently, as you've been following the news about measles. But epidemiologists and public health folks have established a set target percentage of the population that needs to be vaccinated against measles in order to obtain herd immunity. It's about 95% of the population. That's kind of mathematically what we need to achieve in order to protect the population from this vaccine-preventable disease. Certain pockets in the country have fallen below those levels of vaccination, and because of that, we get transmission of this disease that would otherwise not be such a big problem. So that's sort of what happened out in West Texas and Lubbock and other places recently where we've had not quite unprecedented measles outbreaks, but significant measles outbreaks so far in 2024 last year.
I have two questions.
Yes.
The first one is, when you mentioned snake venom earlier, how come we can't prophylactically vaccinate things? Like, it's always after the fact. Snake venom, spider bites, things like that, why can't we just be immune to it?
My guess is it's a matter of economics and scalability. Yeah. Snake, I mean, the one thing, I've never been bitten by a snake, but the one thing I know about antivenom is it's expensive, it's hard to make, and it's hard to get too. Like, you gotta hope you're in the right place if you get bit by a snake and you need antivenom.
When we were going out to Africa years ago, one dose was like $30,000. Oh shit.
Okay. Okay, it's cost.
Yeah.
So not worth my life, got it.
Yeah, well, didn't say that. But it's like, when you do the risk assessments and you calculate not just like how severe the risk would be, but then like, if you're getting bit by a snake. If you're like a homebody like me, it's 0%. Not worth vaccinating.
My second question, passive immunity.
Yeah.
I'm assuming there's more examples. Saliva has antibodies.
It does, yeah.
Sperm, body fluids. Can you transfer antibodies through sex?
Yeah, maybe. But I guess my thought would be, let's just stay with saliva for a minute. Easier to concentrate on that. So you do have a lot of antibodies in saliva, because it's a secretory surface, like if you, the buccal tissues in the inside of your mouth, for example, and the parotid glands that are secreting a lot of saliva. It has a lot of antibodies in it. If you're swapping saliva with someone, will there be antibodies that trade off between you and that person? Yes, for sure. But how long?
Significant enough.
Yeah. So even if you get a mega dose of that person's saliva, those antibodies may not gonna last very long. If you contrast that with the like mother and fetus example, that fetus hang out there for a long time, for months and months of continual injection is not the right word. Exposure to maternal antibodies, like they're just kind of trickling across that barrier. So, and even after they're born, when they're nursing, that's like a continual exposure to those antibodies. It's a significant enough source of those where they're protected for a long period of time. But it's an interesting concept. I've never thought about it before. I just think that those antibodies would be destroyed pretty quickly. They just get rid of. They're just fragile little proteins. Yeah. Good question. Okay, so herd immunity we've defined. When we talk about disease eradication, that is the holy grail of public health. We'd love to eradicate diseases, and it is possible. We have precedents for disease eradication, but it is exceedingly rare and exceedingly hard. Really, the only one we claim total credit for is smallpox, which we eradicated in the late 70s, early 80s. We stopped it from circulating in the human population. We were able to do that because it's a couple of reasons. It's a human-specific virus, so we don't have any zoonosis, we don't have any spillover risk from animal populations. We had people who were willing to get vaccinated, believed in the merits of the vaccine. We had people that were afraid of the disease, which we seem to have lost some of our fear of some of these diseases. So because of that, we had enough immunity as a population to break transmission chains and clear that virus from circulation entirely. Stopped it, it no longer exists. The only reason why we, as military people, sometimes get vaccinated against smallpox, anybody want to hazard a guess? Because it could be used as a biological weapon. Yeah. And because we would ostensibly be the ones targeted for that. But because we stopped vaccinating the general population a long time ago, if we had a biological attack with smallpox, most of the population would be extremely vulnerable to that disease. Because we do not have immunity to it anymore, unless you were born before the 70s. Other diseases like polio, we are getting close. There's still like a little pockets of polio, like wild type natural polio virus circulating in different parts of the planet. But it's like a slow burning fire where it's been difficult to stamp out those areas. some areas where we still have circulating poliovirus. But everyone in this room and general population alike, we all get vaccinated against poliovirus. In this country, for sure. But a lot of diseases, that's simply not feasible. It's not a reasonable goal to eradicate diseases for a lot of reasons, but those are exceptions. Other benefits of vaccination, we have reduced healthcare costs, very pragmatic. If you could actually get people to take vaccines for all the vaccine and preventable infectious diseases we have, that would be a great thing for healthcare costs. It would bring them down substantially. In terms of types of vaccines, we'll go through this relatively quickly. We have live attenuated vaccines like measles, mumps, rubella, and the chickenpox vaccine. So this is the actual real deal. These are both viruses. So in this case, when we say live virus, we mean it's not a living thing, but it is competent, able to replicate, but we've weakened it so much that it's no longer going to be at risk for causing serious disease. Live attenuated. We also have a formulation of the flu vaccine called Flunas, the quadrivalent Flunas vaccine. That's also a live attenuated vaccine. We have inactivated vaccines that have killed virus or bacteria. One of the polio vaccine formulations involves a killed virus. Hep A, that's another virus, that's a killed virus vaccine. Those tend to be pretty good vaccines also. Not quite as good as live attenuated ones. Then we have the subunit or recombinant or polysaccharide or conjugate vaccines. These are usually considered to be some of the worst vaccines in terms of stimulating immune response. And that is because your immune system only gets exposed to a relatively small piece or portion of that microbe. This is probably like with hepatitis B, for example, that's probably surface protein that has been modified and formulated as a subunit. That's probably why it's not super immunogenic. We also have some polysaccharide vaccines, like for strep pneumonia, we have a polysaccharide vaccine. So your immune system is forced to work with a carbohydrate target, which is kind of a tricky thing. Toxoids, inactivated, the use of inactivated toxins, that's what a toxoid is, an inactivated toxin produced by a pathogen, tetanus, diphtheria, cholera, anthrax, all those things. mRNA vaccines, we have had the technology to make mRNA vaccines for a long time. It wasn't until COVID where we finally got that push to start using them. So both the Moderna vaccine and the Pfizer vaccine are mRNA vaccines. Those basically just encode in nucleic acids, in messenger RNA, the ability to synthesize a viral protein, like spike protein. Something super immunogenic like that. And then finally, we can use viral vectors as well. These are pretty good types of vaccines. We have viral vectors we can use to vaccinate people against Ebola virus, scaffolded on a totally different virus called adenovirus. So we put the glycoprotein of Ebola on a different viral scaffold, and it makes for a safe vaccine that triggers pretty good immune response to that. With this summary table, you don't have to memorize this, but the only column that really gives you anything new is this column on the right that tells you about the efficacy. So if you want to just take a look at that at some point, see if you can make sure that that sort of makes sense to you. Like subunit protein-based vaccines, as I said, it's relatively poor. They might not last a long time, and you might need frequent boosters in order to sustain that immunological memory. Okay, you guys doing okay? You guys need a short break? Yes, sir.
I just had a quick question on SARS-CoV-2 vaccine. Was it Johnson & Johnson? I know that wasn't an mRNA. Was that a subunit or
I don't think it was a subunit one, but I don't remember exactly. Yeah, there was, for like this list here, or on this list here, for COVID, it was like everything under the sun. We had viral vector vaccines, mRNAs, subunits. I don't know if we ever developed or marketed live attenuated or inactivated. Other countries did, but we had a whole panoply of different pharmaceutical companies developing different types of vaccines for COVID. Yes.
For the viral vectors, what's that genetic material? It's not the mRNA or
So
what
it's
have you?
So it's the nucleic acid in part, and it's the rest of the viral structure from a different virus. A lot of times we'll use a virus, if you've never heard of adenovirus, that's probably the most common scaffold virus that's used to vector immunogens for vaccine developments. We can use lentiviral vectors, blanking on some other ones, but like adeno is the first one that comes to mind. Okay, oh, yes, sir.
Another question I had, the reason we see the overdoses for the Massachusetts is because multiple goes back to
Yeah, how many of you guys have the anthrax vaccine? A lot of people. How many dosages did you get? A lot, kind of, yeah. And like another military specific niche vaccine that the general population doesn't get, because do you think the general population would tolerate a 6-dose vaccine? You can hardly get them to take a 2-dose vaccine. So, but the military, you can make them do whatever you want. Right? It's wonderful. Okay, so disorders of the immune system. This is just a small little primer on a very big topic. I think some of this you'll pick up with other courses down the road, so it's just kind of like an intro to some of these things, but getting at the water's edge of this course. A couple different categories of how we define disorders of the immune system. The first really isn't a disorder of the immune system at all. It's something you acquire from really nasty pathogens like Staph aureus. It may not seem like it can do much, but it can produce antigens called superantigens that can cause such catastrophic dysfunction and dysregulation of your immune system that it can be fatal. So we'll talk about that, like an acquired dysfunction. Autoimmune diseases, it's a little bit outside of our wheelhouse, but we'll touch upon that since we're talking about the immune system. Immunodeficiencies, anyone think of an example of an immunodeficiency? Yeah, HIV causing AIDS. That's an acquired immunodeficiency, but there are plenty of genetic immunodeficiencies as well. And then hypersensitivity reactions, where your body is basically forming an inappropriate immune response to something it shouldn't overreact to. An innocuous antigen, a benign little antigen, maybe a food allergen, perhaps. And for some reason, your immune system is just not recognizing it. And then with immunodeficiencies, this cartoon is basically just showing that you have some pathogen that should not be a problem, that is becoming a problem because you have a blind spot in your immune system and you're not able to clear it. And then autoimmunity, where you're recognizing self-antigen. Okay, so the first we'll talk about is by far the most important for this course. We'll talk about superantigens a lot in Block 3, bacteriology. And what they are potent bacterial toxins and viral proteins that bypass normal antigen processing, and they result in a massive release of cytokines. So this non-specific binding that occurs, where should the antigen be in this cartoon normally if you have an antigen presenting cell that is showing an antigen to a T cell? It should be in the middle of that cleft. That's where it belongs. And with superantigens, it is not binding specifically as it should. It's binding on the outside of that interaction. And what that's doing is causing the messaging that's going on between the antigen presenting cell and that helper T cell to go haywire, go absolutely berserk. It's causing like an uncontrolled amplification of cytokines that are being released and stimulation of that CD4 T cells. Stimulated CD4 T cells do what? What's their effective function in the first place? Not antibodies, but they're messengers, right? They're the command centers, the things that orchestrate and coordinate the immune response by releasing cytokines. So if they get overstimulated, they go crazy with releasing inappropriate pro-inflammatory cytokines like TNF alpha, IL-1, IL-2, IL-6, interferon gamma, all these cytokines that are just let loose. It's sort of like being electrocuted, and you can't lessen that signal. So because of that, we have some really serious consequences that happen. You get cytokine storms, which we'll talk about a little bit more on the next slides, but all these pro-inflammatory cytokines ultimately lead to things like severe systemic inflammatory responses. We have this uncontrolled positive feedback loop with amplifying signals, autocrine signaling, paracrine signaling. We'll talk about how that can become life-threatening soon, but basically just causes massive dysfunction, first at a cellular level, then at a tissue level, and an organ level. And in terms of the number of T cells that get activated as all these cytokines are being released throughout the body, what normally happens during an infection is you have 0.001 to 0.001% of T cells being activated. If you have a super antigen, 4 to 6 log increases of T cells are activated, up to 20% of your T cells. So that is an incredible uptick in activation, and it's something that is just very, very difficult to treat clinically if you have a patient who has this much overstimulation of the immune response. Okay, in terms of things that can trigger cytokine storms and act as super antigens, with the bacterial threads, we have both gram-positives and gram-negatives that can trigger these different uncontrolled responses. With LPS, LPS is a lipid polysaccharide in the cell wall of gram-negatives, right? We talked about a component of the outer membrane of gram-negatives that had a really, really nasty immune signaling molecule. You guys remember what that was? Lipid A, which has what kind of activity? Endotoxin activity. So with gram-negatives that have LPS with tons of endotoxin built into that membrane, if you have the release of that endotoxin into the system, what you get is a huge stimulation of toll-like receptor 4, which recognizes LPS, and you get huge kickoff of these pro-inflammatory cytokines. With gram-positive bacteria, the lipoprotein acids in the cell wall there similarly can stimulate toll-like receptor 2s, TLR2s, and those too can stimulate these pro-inflammatory cytokines to be expressed. In conjunction with complement, those two together can cause issues like all these pro-inflammatory issues, vasodilation, diathesis, leaking blood vessels, hypotension, because your blood vessels are no longer able to sustain blood pressure. When that happens, you cannot keep organs properly perfused with oxygen, and those organs start failing. So you get kidney failure, you get liver failure, you get brain failure, you get heart failure. You also get DIC, disseminated intravascular coagulopathy, this massive clotting disorder that again affects your ability to maintain blood pressure. These things, when they start combining organ failure, loss of blood pressure, all these things together, you start going into septic shock, then you go into a coma, and then you die. So all these factors together, and all of this just because you have uncontrolled response to some of these pathogenic triggers in the immune system. This slide I don't want to say too much more about except for the fact that TNF-alpha is a really potent player in cytokine storms specifically. It's a pro-inflammatory cytokine. It is necessary, but like all good things in moderation, right? Low amounts of TNF-alpha do good things like contributing to recruitment of neutrophils into that area and clearance of those bacteria. So it's a great and wonderful thing. Activating macrophages, neutrophils, all very nice. Even the systemic responses you get can be normals. The black arrows indicate normal and good positive benefit from TNF-alpha. So systemically, some fever, those pyrogens get released. Liver damage is not a good thing, but you can find that in the lab in order liver panels and see that kind of damage reflected in systemic responses. When you have high quantities of TNF-alpha, not only do you get the local responses and the systemic responses, but that's when you get those really dangerous septic shock conditions. Okay, so those are acquired dysfunctions brought upon by overreaction to like legit microbial pathogens. Let's talk about some actual disorders of the immune system. When we have autoimmune disorders, this is when some of those immune cells that we've talked about fail to develop the ability to distinguish between foreign and self. And as a result, you get inappropriate attacking of self antigens in the body. A couple different mechanisms that can happen. One is through molecular mimicry, where it's really no fault of your cells, but they are attacking something that looks an awful lot like something they've been trained to identify as bad. Molecular mimicry. Some pathogens actually take advantage of molecular mimicry. Like some parasites will coat themselves with antigens that are self-antigens that look like human antigens. And because of that, the well-trained immune system will just ignore it because it looks like self. So molecular mimicry is a common theme, but that can also cause problems if you're attacking self-antigens. Bystander activation is an infection-induced inflammation and tissue damage that causes release of self-antigens. With some cases like endocarditis, for example, you can have damage to the heart that releases molecules like cardiolipin. Cardiolipin shouldn't be released, so when your body sees cardiolipin from damaged heart tissue going into circulation, you might raise antibodies against it, and as a result, your own immune system starts attacking heart tissue. So that's an example of this bystander activation. And then finally, loss of tolerance, where you can no longer eliminate self-reactive lymphocytes. We talked about this a lot with T cells when they're positive selected and negatively selected, and all of that happening in the thymus. If something doesn't go right in that process, where your T cells are being either down-selected or up-selected, what can happen is those T cells get out of the thymus, they go to the lymph nodes, and they're not properly trained, so they start attacking self-antigens at that point. That's why it's so important that that system overall. We will talk about all of these examples in block 3 in a little bit of detail, but I wanted to at least set it up in this block of immunology so you see it first here. Streptococcus pyogenes is a gram-positive bacteria. It can cause, strep throat is probably the most common and well-known problem that it causes. But as a sequela, it can also stimulate your immune system to start attacking your own tissue inappropriately, and that is called rheumatic fever. You haven't heard of rheumatic fever before. Another example of an inappropriate response after a bacterial infection is Guillain-Barré syndrome caused by Campylobacter jejuni. Campylobacter jejuni is an enteric pathogen. It's not the only pathogen that can stimulate the body to start attacking itself, but it's probably the most commonly implicated infection that causes Guillain-Barré. And what Guillain-Barré is simply when your antibodies start recognizing myelinated nerve cells, and it'll attack that myelin layer, that sheath over nerve cells. So as a consequence, you get dysfunction in transduction of signaling through the nervous system. And then finally, reactive arthritis. There's a bunch of things that can cause reactive arthritis. Chlamydia trachomatis, like STI form of chlamydia trachomatis, Shigella, Salmonella, enteric pathogens, that can all cause unintended, inappropriate immune targeting. Immunodeficiency orders. As I mentioned, there are some important genetic immunodeficiency orders. Too many to count, too many to enumerate, and maybe, hopefully, you guys will get that in another course. But suffice to say, imagine everything we've talked about in this immunology part of this block. If there is a problem in any one of those immune cells function, things just fall apart. This whole system, I hope I've managed to get that point across. This whole system relies upon other members of the immune system in order to work properly. So if you have a T cell dysfunction, that's not going to just affect T cells. It's going to affect all the cells that T cells are interacting with. If you have the inability to produce cytokines or even cytokine receptors, that causes massive failings in other parts of the immune system. So these genetic immunodeficiency orders can be devastating. They can make patients really, really susceptible to disease. And then we also, of course, have acquired immunodeficiencies, which we'll talk about a little bit with HIV. And it's this really malicious way it has cells and just depleting those over time. So the body can no longer respond to any infection. It makes it extremely difficult for the body to fight off infections that we would normally have no problem fighting off. So those are called opportunistic infections or aids to fighting illnesses. You sometimes hear that term as well. You don't see it in healthy people. Finally, hypersensitivity reactions. I think this is the last slide of the deck, yes. Okay, so hypersensitivity reactions are also exaggerated or inappropriate immune responses. This one example that I put here is actually a beneficial hypersensitive reaction. And this is kind of like the response we get when we are screening people for exposure to tuberculosis using the tuberculin skin test. You guys have probably all had this test before. Yeah, PPD, purified protein derivative. So this test, if a person has been exposed to tuberculosis au naturel, or if they've been vaccinated with BCG, either way, they will have sensitivity to the antigen that's in that particular formulation. So it's a tuberculin antigen, you're putting it under the skin, just a little bit under the skin. And if that person has encountered Mycobacterium tuberculosis before, they have a cell-mediated response to that, and you get localized inflammation in that area. It's not a big problem. It's not going to cause any significant pathology, but because it's a delayed hypersensitivity reaction, that person is asked to come back 48, 72 hours later so that hypersensitivity reaction can be read. And if it's positive, you get a large induration in that area that you can actually measure and see if that person has been exposed. But what that induration is like an infiltration of immune cells into that area, like the cell-mediated immune response. So mostly T cells that have previously been activated and exposed to tuberculosis before. That's what this cartoon is showing you, that induration that forms within a day or two or three. Creates that induration. So I'll leave it at that. We could talk about this in a lot more depth, but I think that's all I want to say for you guys. Yes, sir.
I've been persecuting too deep in the weeds, I don't know. For cytokine storms, does the super antigen, is there anything specific that causes the super antigen to bind outside of the proper site to create that, or is it just one?
That's a really interesting and good question. So at the very bottom of this slide, examples of sleeper antigens. I haven't studied these examples, like the enterotoxins that are produced by Staph aureus and toxic shock proteins, toxins, in enough detail to know if, like, structurally, there's something about them that is conducive to that inappropriate binding. But yeah, that's something I'll have to look into. I'm not aware of that on a structural level. Great question. Any other questions? Okay, so the plan that we've worked out between now and next Friday the 30th, when micro exam 1 is that we're done with the lecture material. Ergo, on Wednesday's lecture at 1300, we're just going to cancel that unless you guys want to have a lecture, but there's nothing really to talk about. We're done with the material. So the plan that we've talked about is we'll just do a review for exam 1 on which day? Monday? Tuesday?
Tuesday after the A&P test.
Tuesday after the A&P test. Okay, so that'll be after 2 exams from now. You'll have biochem and another A&P exam under your belt. So at that point, on Tuesday when we meet up, you can just focus entirely on micro, and you can bring all your questions. You can let me know what is confusing to you, and we can just hit it hard on Tuesday. Then you'll have another 72 hours to study for the exam. Sound like a plan? Okay, cool beans. Yeah, so I won't see you again. Let me know if you want to meet me for office hours. I'll be in Tuesday and Wednesday this week. And otherwise, I'll just plan on seeing you guys Tuesday. So have a great weekend. Good luck on your exams.